Right, hello. There won't be many people on this one because I um, sent the email very late because I forgot that the link has changed. So that's what it is. Um, this might be a little bit of a problem because if you come on a Saturday usually, then we will have done different things than if you come on a Sunday, just because of the day in which we change topics. But if you have anything to say to me, then feel free to do so first. Otherwise, I will pick some questions at randomish to go through. Um, I please show it to you. Um, what day do you come? You come on a Saturday, don't you? So you will have had not a booklet technically, but an exam paper like this one, which would have been a booklet, but it wouldn't have been one of ours, if that makes sense. Um, so on Saturday you'll have done biology, and Sunday we did chemistry. So it's all messed up. So anyway, well, you got given basically a foundation level exam paper because it's all got topics that we have done this year, and it was all a mixed lesson of random things. A lot of which you won't, some of which you won't know, but it's just good to practice an exam paper. I might end up going through most of this, unless somebody has any questions on the Key Stage 3 stuff. Um, 4 to 8, 4 to 8. That's going to be so long. Um, all of these. Okay, fine. I'll go through this first. If you have anything else that you want me to do, then please tell me. I will be here till about 8-ish. So if we have other things I can do, um, as well. See who else is here. Just send those topics again because I don't remember what they were. Just send them again, or at least tell me here now what those topics are. I think they were. I have a feeling they were chemistry paper two topics. I feel like they were, but and if they are, I'm going to suggest you do something else. If they are chemistry paper two topics, instead of doing much of it today, I'm going to tell you another suggestion. I'll find what they are. Physics paper two topics. Um, waves in. Okay. Yeah, I can certainly do that required practical. The acceleration one is just that thing that moves. And yeah, I'll do some of the separate topics. But what I might do is suggest to you, if you are available, on Sunday evening. I'll tell you on Sunday when I see you. But on Sunday night, because physics paper two is on Monday, as in the real one, I will be doing an extra thing with all our year 11 students in going through all of those things. Anyway, like in detail. So that might be a useful thing for you to join, which I can tell you about. In fact, to be honest, I can just tell you now, it's going to be on the link that you're on right now. Because so if you want to join that on Sunday, it's at 9 o'clock on Sunday, which is quite late, but that's when I have to do it. But I will be going through all of that stuff, but I'll do a little bit of it now. I'll go through these questions, given that nobody else is asking me anything else. Now, and then I suspect some other people might turn up as well soon. So, questions 4 to 8 on this paper, which is a biology paper. Bear in mind, I should also tell the few of you that are here that you will have a test in your first lesson in July, which is why the remaining lessons you have are just going to be like revision of stuff we've done this year, in order to be able to do that. But it's going to be on basically every, in theory, it's on everything, but it's not really. Um, okay. Right, questions 4 to 8. 4 to whatever you said. 4 to 8, yes. What was the student B's resting heart rate? This is how exercise affects heart rate. Obviously, when you exercise, your heart rate goes up and then whatever. Student B is the dotted line. Their resting heart rate is what it starts at. So you have to just read off this carefully. It's going up in twos, so that is 66. They started running at 2 minutes. What evidence is this there in figure 4? So at 2 minutes, what happens to both these graphs? The heart rate starts to increase. So that's all you want to say there. The heart rate increases. How many minutes did the students run? Um, well, presumably they stopped running when their heart rate started to come down, right? That's when you know you stopped running. So if we started running at 2 minutes and stopped running at 6 minutes, have we not run for 4 minutes? That seems to make sense. Um, Unfortunately, the biology paper two session has completed because biology paper two was on Monday. So I did that last Sunday night. Obviously, we haven't invited you guys to it, or if you're in year 10 and stuff, because it's not specifically for you, but just for your own benefit, just so you know that if you are with us in year 11, you get quite a lot of stuff in the lead up to your exams, which is why I've been very busy in the last couple of weeks doing lots of extra stuff. So yes, in terms of what's left for our students, there is just a chemistry paper two and a physics paper two, for which I do always a night before revision thing. So chemistry's on Friday, so Thursday night I'll be doing that. And on Sunday night I'll be doing it for physics paper two. Basically, that's what I'll be doing. How many minutes? Well, I think we just said four. Student B is fitter than student A. Use figure four to give two pieces of evidence to support this student. So, student B is fitter than student A. How do we know this? Well, because firstly, his maximum heart rate was not as high. If your maximum heart rate is not as high, it's mainly because it's partly because you're fitter. So his maximum heart rate was lower than that. Also, look how quickly the heart rate went back to normal, right? It took quite a short amount of time to get back to normal, whereas for A, it took longer to get back to normal. See what I mean? It's taken a lot longer to get back to normal. And so that's another thing. You could also mention the resting heart rate. Your resting heart rate is a sign of how fit you are. The lower it is, the fitter you are. So student B has got a lower resting heart rate than student A as well. So there's 3 points there that you could say. You could say that student B has a lower resting heart rate, the maximum heart rate was lower, and you could say that it returned to normal quicker for student B than it did for student A. Other changes in the body during exercise. Why do these changes occur? Why do things happen in your body, basically, when you're exercising? Well, this is a very common question, but it's often phrased in lots of different ways, but the answers are virtually the same. The kind of things that happen when you're exercising is obviously you breathe more heavily, right? So you can mention that your breathing rate increases. Just want to put arrows here. Um, and you could also mention that you sweat, you get hotter. Like there's quite a lot of things that you happen to you. You're hotter, you sweat more. But why do all these things happen? So why does your breathing rate increase? The reason your breathing rate increases is because what you're trying to do is provide more oxygen to your body. Why do you need more oxygen? Because it's for respiration. And what does respiration do? It releases more energy. Okay, so you can mention that probably on its own is 4 marks, but if you want to say some other stuff as well, then you can say that why do you get hotter? And also, why do you sweat, for example? The reason you sweat is because your temperature increases as you are respiring. So respiration actually makes you warmer. And then why do you sweat? You sweat so that you can cool yourself down. So that's another sort of few things that you could say for that. But I think the best answer really is around respiration and energy, um, so that you can exercise more, right? That's why you do it. 5, when an organism grows, new cells are produced by cell division. What type of cell division happens to produce new body cells? It's called mitosis, which I appreciate. Again, some of these things are a little bit advanced, and also some of them we haven't specifically done because we leave certain topics in what we do in the next year, and mitosis technically is one of them. Why can cancers grow very large? Cancer is an uncontrolled cell division. It's when it's uncontrolled. A cell, one of your cells basically mutates and it starts to uncontrollably do that. And it starts blocking things and it causes loads of problems. And so that's actually what cancer is. Who's here? Nobody else. Fine. Give one factor which increases the risk of getting cancer. You could have mentioned smoking. You could have mentioned eating high fat foods. You can mention exposure to radiation, smoking, alcohol. These are the easiest answers, I think. Um, Survival rates for people with cancer have improved a lot. People who are alive 10 years after diagnosis are usually considered to be cured. People who are alive 10 years after. Okay. Figure 5 shows data for people diagnosed with cancer in 1961 2001. Okay. Always spend a bit of time looking at graphs when you do these things. So it tells you that in 1961, that's a darker bar. Breast cancer, 40% of people were alive 10 years after diagnosis. With prostate cancer, there was this one, but as you can see, in 2001, 10 years after diagnosis, a lot more people were alive, which suggests that we obviously figured out ways to treat cancer and stuff over the years and are better at keeping people alive. 78% of people diagnosed with breast cancer in 2000 were alive 10 years later, complete it. So 78% of people, there's one bar missing here that you can see here, this one. And that basically told you it's 78% for that bar, so you just have to draw it. 78% goes up to about here, so my bar will look something like this. Something like that. Which type of cancer diagnosed in 1961 has the highest survival rate? So the 1961 is the dark bars. Which one's the highest? Testicular cancer. Looks like it to me. Which type of cancer shows the biggest improvement in the percentage of people alive after 10 years? So which had the biggest improvement? That looks like prostate cancer to me. So improvement means the difference in the two bars. So that compared to that, obviously you could check the numbers precisely, but that looks to me like the biggest difference from what it was to what it is. And the answer is that one. Two reasons why the survival rate for all cancer have increased. So why has this happened? Why is it that we have that? You could mention improved treatment. So we've got better medicine and treatment things. You could also mention improved like patient lifestyle and things. Like generally people smoke less or eat better a little bit more compared to they might have done before because of more education. That can be an answer. You can also just mention earlier diagnosis. Like we are picking it up earlier now, therefore more people are likely to survive it because we get to it earlier. So I want to mention that one. Okay, to 8, you said, right? So I'll get through this. Pathogens cause infectious disease in animals and plants. Draw one line from each of the type of pathogen. Again, you'll have to learn these. You'll have to learn that gonorrhea is caused by a bacteria, malaria is caused by a protist, measles is caused by a virus. These are very specific. There's only like two, well, actually, that's not true. There's about 6 or 7 different diseases, but you need to know which pathogen they relate to. So that's an example of that. Some parts of the human body have adaptation to reduce the entry of live pathogens. How is the trachea adapted to reduce the entry of live pathogens? You are meant to know that the trachea has mucus in it, which traps pathogens. It's the sticky stuff, right? And you can mention something else. There's also these little hairs called cilia, and what they do is they move the mucus back up your throat so that you can spit it out or swallow it. Um, and that's how you get 4 marks. Okay. Malaria is seriously fatal. It's spread by mosquitoes. Scientists investigated the behavior of mosquitoes to understand how the spread could be controlled. This shows the equipment. So in container A, they had some infected mosquitoes and some socks worn for a day. And there was container B, which had uninfected mosquitoes. The hell? They 30 mosquitoes were placed in A, 30 in B. The number of times they landed on the socks was recorded. Okay. Bloody hell. So weird. Name the dependent variable and the control variable. The dependent variable is the thing you're measuring. So what you're measuring is the number of times they landed on the sock. That is a dependent variable. Number of times landed on the sock. And then control variables, you have to think about control variables a bit more, but they are things that have to stay the same every time you do the experiment. So things that stay the same will include the number of mosquitoes. Often there's a clue in the description of the method where they mention we use this amount or this whatever. So the number of mosquitoes would definitely be an answer. Also, it mentions socks worn for a day. You would need to make sure that's exactly the same amount of time that they've been worn for. Yeah, okay. Um, and then that would be the two best answers. You can say other things like the temperature or the type of socks or how long you do this for. Time is important. How long you do this whole experiment for is important. It's a weird one that. So I presume the assumption of this is that mosquitoes are more likely to land on one of them than the other. Yeah, okay. Um, so infected mosquitoes and uninfected mosquitoes. So infected ones perhaps are more likely to try and bite things. Um, Infected mosquitoes landed on the socks 3 times more often than uninfected mosquitoes. How can this be used to reduce the spread of um, malaria? Well, um, oh God, that's a difficult question. So, the idea of this, the knowledge you're meant to know of this, is that one of the ways to reduce malaria is if you could somehow get rid of mosquitoes, right? If you could catch them and get rid of them, okay? And so, what you could say here is, or you, yeah, and so what you could do here is if you know now that these socks attract mosquitoes, what you could do is go, right, what I'm going to do is use these like as a bait and get these mosquitoes near some socks and then um, trap them to try and kill them. Essentially, we could use it as like a trap or a lure to them. So we now know that they get attracted to these things. So if we can get some socks, put them somewhere, get those mosquitoes attracted to them, and then, I don't know, kill them somehow, then that would reduce the spread of malaria. So you could say something like that. There are a couple of other answers there. But so you may use worn socks to attract and trap mosquitoes. And what that would do is reduce the spread of malaria because it only gets spread if they go around biting people. So if you can catch them somewhere, then you're going to minimize that problem. Right. Tobacco mosaic virus is a virus which basically makes green plants more yellowy because what it does is it destroys the chloroplasts. This is a very classic question about this. Why would having fewer chloroplasts affect the growth of a plant? Well, if you have less chloroplasts, it means that you have less chlorophyll, because chlorophyll is the stuff inside the chloroplasts. What does chlorophyll do? It absorbs sunlight. And so if there's less chlorophyll, there'll be less light absorbed by this plant. What is the light used for? Photosynthesis, surely. And so there'd be less photosynthesis. This is how you do these questions. You have to sort of take it almost like a ladder, as if you're going through each step and explaining the consequence of each step in turn. And that's how you start building up the number of marks that you're getting. Less photosynthesis, and then that's enough actually for 3 marks, but you can then go further and say, well, what does less photosynthesis mean? It means the plant makes less glucose, doesn't it? Because that's what the whole point of photosynthesis. And that glucose is used to help it grow, so it's not going to grow as well. So just from having less chloroplasts, we can explain exactly why that leads to the plant not growing very well. It's because it's not going to do as much photosynthesis, essentially. Um, if you are here from the year 11 stuff, I'm probably not going to be doing any. I might do a tiny, tiny little bit of physics stuff, just so you know, paper two stuff in a minute. But otherwise, we'll be doing it on the day before thing. What is the correct word equation for photosynthesis? It is. How is it again? A glucose on the other side. So that one. That is the word equation for photosynthesis. Always look for glucose and oxygen together. And if it's photosynthesis, they should be on this side. If it's respiration, they'll be on the left-hand side. So actually, that is respiration, just so you know. This shows some of the apparatus that we use to measure the rate of photosynthesis. This is a required practical, which again, we don't specifically do until later in our normal lessons. Just wanted to ask in advance. Can you go through NPK fertilizer? Yeah, of course. Question 5C and 6B on the year 10 chemistry book. Yes, I'll do that as soon as I've done this bit. Yes, that's fine. Tomorrow I will be going through NPK fertilizers with the year 11 stuff. No problem. Um, just remembering what each one does. Right. Jeez, how could you investigate how the rate of photosynthesis is affected by different colors of light? What you would measure and variables you would control. Okay, so if I wanted to find out how light affects the rate or different colors of light affect the rate of photosynthesis, I would get a lamp and place it a fixed distance from the pondweed. So place a lamp a fixed distance from this thing. So you never change the distance because you're not investigating how the distance or the light intensity affects the rate of photosynthesis. You're just going to change the color of the light each time. So you're going to place the lamp fixed distance away and then count the number of bubbles produced in, and make sure you specify a time. It doesn't matter what that time is, but let's just say in 1 minute. It doesn't really matter as long as it's the same every time. Count the number of bubbles in a minute. Then what I might do to investigate how the color of the light affects that, I might change the light bulb to a different colored light bulb. Okay, change the color of light and repeat. That's what we're going to do. And you'll do that with different colors of light. You can probably get marks for saying repeat with each color and calculate a mean. So when you do it with, let's say, red light, do it 3 times and calculate an average of the number of bubbles because it avoids anomalous results. And then that's it really, isn't it? You can then compare which one gave you the most bubbles, which color of light. But they also said variables you would control. We need to make sure that we keep this distance the same every time. That's why I mentioned it. Fixed distance. We should always use the same type of pondweed. Um, that should stay the same. We should also do this for the same amount of time, which is why I specified the amount of time. So don't do it for 1 minute for one and then 3 minutes for the other. That doesn't make any sense, right? Because you're going to get different numbers of bubbles and things. Um, so always do that. Right, I think that covers that mostly. Mm. So let me just make sure. 189620328. Okay, right, that was done. And then the scientists said light, so they drew a graph basically. With a similar investigation, not the same one. And all they did was change the light intensity. Always make sure you read these questions really carefully because they change things suddenly. So now what they're investigating is how the light intensity affected it. And it seems like as you make it brighter, the rate of photosynthesis increases, but then it levels off. The scientist said light stops being a limiting factor at 20 units. Give evidence to support this statement. Yes, that's true because the graph has flattened out, and that shows that light is not the limiting factor. What could be the limiting the rate? So there's only 3 things that affect the rate. There's light intensity, carbon dioxide, and temperature. So one of the other two, like temperature or CO2, will be the limiting factor. And then last question for this one. Oh God. Amylase is an enzyme that digests starch. We investigated the effect of pH on the activity of amylase. They put some, again, this is going to be hard because we haven't done the required practical. It's more for you to just get some experience of seeing these. Amylase and starch in a test tube. They put it at 25 degrees. Every 30 seconds, they tested it for the presence of starch. What's going to happen over time is that the amylase will digest the starch. So eventually it will all disappear. And what I can do is see how long it takes to disappear at different pHs. You're meant to know that amylase is an enzyme. All the enzymes, anyway, work best at a certain pH. And you can see here that at this kind of pH, it took the least amount of time for that starch to disappear. That means the amylase was working really quickly. It took only, 1.5 minutes to make it all digest, whereas at pH 5, it took ages because it doesn't work as quickly at those pHs. They concluded that 7.25 was the optimum. That is not valid. Why? Well, firstly, 7.25 wasn't a pH at which we did this experiment, so we don't know. Secondly, it was both the same at 7 and 7.5, so we don't know where it would be in between those two values, would we? Um, so we've got the same result at 7 and 7.5. So how do we know it's 7.25? And we haven't tested it at 7.25. Also, it's not very valid because we never repeated the experiment. We should be repeating it with any required practical. They did another investigation. They measured the amount of sugar produced every 30 seconds. So what you're meant to remember is that when you digest starch, it breaks down into sugar or glucose. So now what's happening? Yes, the starch is disappearing, but it's turning into sugar. Calculate the mean rate of sugar produced per minute for the first 5 minutes. Well, all you do is you go to 5 minutes, which is there. You go to how much sugar did we produce? We produced 2.6, it looks like to me. 2.6 units of sugar in 5 minutes. So per minute, you do 2.6 divided by 5, and you get the answer here. Okay. Iodine solution is added. Remember what iodine solution does? It is a test for starch, and it goes from orange to black if there is starch present. So if iodine solution is added after 10 minutes and 60 minutes, what would you see? Well, after 10 minutes, you would still see, because you've produced some sugar, but it's still increasing, that means you'd still have some starch present in there. So after 10 minutes, you'd see the starch go from orange to black, or the iodine to go from orange to black, because there would be starch. But after 60 minutes, it looks like we're not producing any more sugar, which means that we've finished all of the starch. So there's no starch left. If there's no starch left, the iodine solution will stay orange because there won't be any starch left. It's all gone. They repeated the investigation at 37 degrees C, so they did this at 25, and now they're repeating it at 37. If you repeat it at 37 when it'll be a warmer temperature, so the enzyme will work faster, you will get a line that looks like this. Remember, very important that you finish in the same place, but you're just getting there quicker by putting your line sort of above the other line. Um, and then that's it. So I'm done. Right, let me do the chemistry question somebody asked me. 5C and 6B on the year 10 chemistry booklet. 5C and 6B. Oh God, yeah. There are a few questions thrown into these which are not necessarily covered in the lessons that we do. And this is an example of a triple science, quite advanced topic. But let's see what it is. I'll tell you. Alcohols are used as fuels. A group of students investigate the amount of energy released when different alcohols are burned. Okay, fine. They show the start temperature. Is this the question? I'm making sure. Yeah, so this is what you said, right? 5C. Okay. Well, the first bit of this is very easy. Right at the start temperature and the final temperature. So the starting temperature looks to me like 22.5. The finishing temperature looks to me like 38.7. And then the increase in temperature is the difference between those two values, which is 16.2, I want to say. And then the student worked out the heat energy released by burning 1 gram of each alcohol. They use this equation. What is the value of M? Is it look at figure 2? What's figure 2? Oh, there, isn't it? Because it's the mass of the stuff that you're burning. So it is 50. And then here are the results. It shows that when we did this with these are different alcohols. Again, this is not necessarily something you're meant to know yet, but there's lots of different alcohols here if they end with ol. It tells you how many carbon atoms they've got, and it tells you how much energy it took to or how much energy was released when we burned them. As you can see, as we go down, we got more and more heat energy. Which value of heat energy is anomalous? It's got to be this one, hasn't it? Because it's not, it goes 11 to 13, then to 20, then down to 16. So that's got to be anomalous. There's something wrong there. Something has happened. What is the relationship between the number of carbon atoms and the heat energy? As the number of carbon atoms increases, what happens to the heat energy released? It increases. That's all you have to say. The value in a data book for the amount of energy released when 1 gram of butanol is burned completely is 36.2, but we got 16.8. So there's two reasons why the student's result is lower than the data book value. Okay, so firstly, it could be that not all of it burnt. Let me see what the mark scheme says. Um, yeah, oh no. Okay, well, I'll tell you the answer though, anyway, that it says. It says that firstly, when we did this experiment, where's it gone? We should have put, when we're doing anything temperature related, we should really use a lid. We didn't use a lid because some of the heat might be lost. We should really insulate the beaker a little bit to avoid heat being lost through the sides. So you can mention no lid, you can mention no insulation. You could even say that we didn't measure it accurately or that we didn't repeat the experiments and calculate an average, so we're not necessarily getting valid results. Any of those kind of things would be sufficient. And then again, you won't know this, but the answer is that one. You will get there soon. And then you also said 6B, which is probably similar. Right. Oh God, I remember this question. It's really horrible. The diagram shows it's trying to demonstrate cracking. As a reminder, what is cracking? It's when we've got like a long hydrocarbon and we're breaking it into smaller, more useful ones like C4H8 and whatever it adds up to, C6H14. See what I mean? The long ones become a bit shorter. Um, ah, right. The things you're meant to know about cracking is that what we need are two conditions for cracking. We need a high temperature and a catalyst. Okay, they're the two things we generally need for um, cracking, which means I suspect that the broken pottery in this example acts as a catalyst because we've already got the warmth. We need something else. The something else is the catalyst. Um, the name of the liquid hydrocarbon produced by the cracking, hey, isn't it? Oh, the name of the liquid hydrocarbon produced is octane. Yes, that's what they want there. And then the name of the alkene produced, the alkene is ethene. Basically all the answers in the thing. Okay. And that's all of that. Right. Let me see who's here. Right, nobody else got any more questions. So what I'm going to just do, just briefly, is talk about a little bit of a physics topic, which someone asked, but as I say to the person who asked it, as a reminder, I'll just let you join on a Sunday when I do more of this. But there are a few um, extra topics, and they're just to be clear for the people who are here, we haven't really done most of these topics because we leave them all till next year. But worth you hearing them because you'll need to know them. No one's asking any more questions. I'm assuming there's no more homework questions now, just to be clear. Um, okay, so as I say, a lot of this, especially if you're not in, and you're not even in year 10, then this will be a little bit um, complicated, but you still need to know all of it eventually. So um, I will start with, there's a required practical, which I will just talk about using my own notes, which you have asked me for, which is all about acceleration. So let me just give you some background before I do this bit. There is an equation, which is force equals mass times acceleration. Okay, which means if you want to find out, let's say there's a car moving along Okay. And I want to find out its acceleration. Acceleration is how much it's speeding up, how quickly it's speeding up. If I wanted to find out that information, then I need two pieces of information. I need to know how heavy the car is, the mass of it, and the force which comes from the engine of the car. So the car engine will provide a force forwards. There will be some other force backwards, like wind, air resistance, and stuff, but overall, this should be bigger. And that will allow the car to speed up. I need those two numbers. I need to know the force with which it's being pushed and how heavy the car is. Then I can work out the acceleration. So clearly, if this is bigger, like let's pretend this, in fact, what we'll do is we'll rearrange this equation to get F divided by mass is acceleration. Okay, so that means that let's pretend that a car was 10 kilograms, just to keep numbers simple. Let's pretend that the force with which it was being pushed was 100, which means its acceleration then surely is 100 divided by 10, which is 10. But if the force was bigger, what happens to the acceleration? Because remember, the car's still the same weight. 200 divided by 10 is 2. Oh, sorry, I got that wrong. 200 by 10 is 20. Um, so clearly, me making the force bigger has made the acceleration bigger. Okay, that's a simple point. We know this to be true because that's what happens. However, what required practicals do, or practical experiments, basically try to prove something you already know. That's kind of how these things work. So we already know that if I change the force on a vehicle, make it bigger, it's going to accelerate more. But how can we prove that? We can prove it using something like this. It's a bit of a pointless experiment, but anyway. Fantastic. Yeah, something like this. So, what you have here, incredibly, is an example of a required practical where you have a trolley, which acts as your car, attached to a piece of string on a pulley. And on the other end of the pulley, we've got some weight. Okay. Now, these things will act a bit like your engine. Okay. So the engine is this. This is the thing that's pulling. Obviously, this is going to go that way. And then this is going to pull the car this way. And so what we can do is do this experiment as it is, let go of this. And what we have here are some light gates. What these light gates do is they're just a fancy way of calculating the acceleration of this as it goes past. They basically time how long it takes to go from one to the other and work out its acceleration, basically. You don't need to worry about how they do that. But what we're going to do is calculate or put some masses on this and then let go of it, and then this will row that way, and this will tell you the acceleration of the car with this force. Okay, so this is my force, this will be my acceleration. Okay, let's pretend based on the numbers that I used earlier that this was 100. Let's say the car is, what did I say it was? 10 kg? 10 kg, right? So 100 newtons of force there. And now I know that the acceleration will be 100 divided by 10, which is 10, but this will tell me the actual acceleration, okay? Because it's like it's connected to a computer, it will tell me. What I will then do is add more weights to this, which means I'll increase that force to make it 200 now. Then I'll let go of it again, and I'll see what will happen. What will happen? The acceleration should be bigger, but the point is I can prove that as I increase the force, I increase the acceleration using this kind of required practical. You often then get asked things that you keep the same. You have to make sure that the mass of the trolley stays the same every time. If you're only investigating how the force affects the acceleration, then it's important, just like I did here, that we keep the 10 the same, right? So we always use the same mass of the trolley. That's pretty much it, really. We can also investigate how the mass effect of the trolley affects the acceleration. In that case, we would just keep this the same, but we would add more weights and make the trolley heavier each time and see what that does to the acceleration. In that case, it will actually decrease it because the heavier the car, the less it's going to accelerate. But again, that can be proved by that formula. So that is what the required practical is in this case. It is in the topic of forces, just in case you care. Right, other topics I'm just going to tell you. Again, this is, I've been asked, I'll just be clear, I'm doing this only because I've been asked about some specific triple science topics in paper 2, which again, we don't do too much of until near the time. But um, God, what happened here? So, waves in solid. I'm not sure what you mean by waves in solid. Other than I can only think of there's a practical involving refraction of waves. Um, because that is what presumably that's what you mean, but I'll just talk about it now. Um, Do the light gates move or do they just detect it? They don't move. They're literally like a light. They just detect the acceleration. That's all they do. There are a couple of ways to do that. You can use a stopwatch and sort of time it for going from one to the next and work out the acceleration. But using the light gates is just a quicker computerized way of doing it. It'll just tell you the acceleration. You don't do any calculations then. But that's why the light gates can be used. They're the sort of most sophisticated way of doing it. But no, they don't move or anything. They're just a light. When you go through one and then the other, it can calculate how long it took, and therefore the acceleration can come from there. Right, I'll just do this little bit because I think it is actually applicable to everybody a little bit, which is I think we'll have to clarify on Sunday what you mean by waves and solids because I think this is what you mean. But waves and solids is just essentially just refraction. Um, which is that when a ray of light or a wave goes from one material into another, especially at an angle, its speed can change. It can often change speed and slow down, especially when it goes from air to glass. And that's why we get this bending of a ray as it goes through a from one material to another. Um, the when it leaves that glass, it speeds up again and it bends the other way. So this is all about the speed changing in terms of this particular practical on refraction. I don't think there's too much else I want to say, but we can be more specific about it on Sunday if you want. I will take your lesson and just have a look at this on Sunday. And we can just go through these topics. But in terms of other things, just so you know, I won't go through this right now, but I will just tell you other things that are in paper two that are if you want to be focused on triple science stuff only. There is a little bit in this momentum. There's something called changes in momentum, which is a formula where it's really interesting actually. It's to do with why we have things like seat belts and stuff. Because it actually delays your stopping. If you just go from moving really quickly to stopping suddenly, it'll hurt. Whereas if you go from moving to stopping gradually, it doesn't hurt as much. And that's why we have things like those safety mats that you might jump on in PE because they delay your impact. And we have seatbelts for the same reason because they're not strapped to you. They actually have a bit of give, and so they bring you to a stop. There's quite a lot of bits around that, which is quite interesting. There's quite a lot in waves, but this bit I'm focusing on a lot, which is there's a bit on lenses that is really rough. I don't know if you need to know it for now. There's a little bit in electromagnetism called the generator effect transformers, and the entire topic of space is technically triple science. So just for you, I will just let you know that we'll chat about it on Sunday in terms of that thing that I'll be doing. And I'll speak to you about, you can just message us about Thursday as well and just join the chemistry one if you want. In which I'll be doing the whole thing, which will be fun. Right, given that nobody else has any more, I won't do any more today. We should be back to, we'll always be on this link, just to be clear, for the rest of this month, and probably into July. And your English lessons are on a different link, just to be clear, but they should be back. They will be back to normal from next week, should you want to do that. But yeah, otherwise, if you have any other questions, then just message us and then we'll deal with it. I'll end it there.